Dear students, I welcome you all to the 16th session of Reproductive Biology and Developmental Biology paper prescribed by the Mangalore University for 6th semester BSc Zoology. In this session, we are going to study about the detailed aspects of fertilization. By definition, fertilization is the process in which it marks the beginning of an individual. It involves the union of two gametes, the sperm and the ovum or egg. In general, most invertebrates and vertebrates accomplish close approximation of spermatozoa and eggs through special devices or particular forms of behavior. This behavior of animals will be there in order to attract the conspecific, the opposite sex of the same species. A detailed approach towards this kind of behavior, what we call in scientific terminology as the courtship behavior, is dealt in the 4th semester itself. So if you want to study about these courtship behaviors again, or to have a fine touch of the courtship behavior, you can go back to the videos that are available in the 4th semester so that you can rememorize the topics that are dealt in your 4th semester. The primary needs for the encounter of spermatozoa and ova are fluid medium for the act of fertilization and delivery of large quantities of spermatozoa close to the number of ripe eggs at the right time. According to the place and nature of fluid media, there are two kinds of fertilizations that have been reported. See, if you look at the detailed process of fertilization, it is not so easy process. It's a very complex process that begins with the identification of the opposite sex of the same species. If at all the successful fertilization has taken place, then either the male or female has to find the opposite sex of their own kind. And then not only that, most of the times the females are very choosy about the father, the speculated father of their next generation. So they go on searching for the fit father, and fitness here is the Darwinian fitness, the ability to survive in a given environment. Hence, a series of rituals which are called as courtship rituals or courtship behavior will be exhibited either by males or females, and mostly it is exhibited by males in order to attract the female. And the female will choose the best father either by observing or by selecting the courtship behavior, the intensity of the behavior being exhibited, or there are different kinds of magnitudes by which the female selects the male. And most of the times, the courtship behavior exhibition will be directly related to the Darwinian fitness of an individual, which offers certain selective advantage over other individuals of the same species to survive in a given environment. The two gametes, spermatozoa and egg, must approach each other before the successful fertilization is possible. Hence, this approximation is only possible in a liquid medium. Based on the place and nature of the fluid media, there are two kinds of fertilization: the external fertilization and the internal fertilization. External fertilization is a kind of fertilization that occurs in a liquid medium outside the bodies of the parent animals. So that means the ovum and the spermatozoa will be released into the external environment. The fertilization takes place in the external environment, development takes place in the external environment. Fertilization, if it is internal, then it takes place inside the body of the parents, and mostly the females, and occasionally the fertilization also takes place inside the body of the male, as in hippocampus. Among the freshwater animals such as fishes, amphibians, and many other freshwater invertebrates, the timing of the spawning of eggs, spawning means the release of egg, by the female and shedding of the sperms by the male parent are very specific, as their spermatozoa remain active usually for only a few minutes. The sperms are directly delivered to the eggs of an individual female immediately after the egg laying. You can see here in these photographs, the fish and amphibians are releasing the eggs. And here you can see a female frog is being held by the male frog very tightly, and this process is called amplexus in amphibians. And once this amplexus takes place, the male will squeeze the abdominal region of the female with the help of its hind limbs. As a result, the female will lay numerous eggs, which you can see here, which are looking like black dots. And upon this, over the surface of this mass of ovum, the male will spray its spermatozoa. And if you could observe one more photograph here, this is the photograph of a fish laying egg. You can see this is the female fish which has laid the egg and is just going away. And immediately after that, the male is ready to spray the spermatozoa over that. See, the problem with the spermatozoa is it can be retained in the external environment. It will live in the external environment only for a few minutes. So before they die, before they become, non-functional, they must be sprayed, or they must be made to contact with the ovum so that the fertilization will be successful. But the marine forms shed eggs and sperms freely into the surrounding water, and most of the marine individuals, such as the echinus, for an example, the ovum of the echinus you can see here, a kind of echinoderm, they release it simply to the external environment. The time interval between the laying of eggs and the shedding of sperm may be even weeks or months because of the saltish seawater, which serves as an important physiological medium for the gametes. During external fertilization, the movement of spermatozoa in a liquid medium is entirely at random, and the spermatozoa collide with the eggs as a matter of chance, which occur regularly in nature, partly due to the enormous number of spermatozoa produced by the male, and partly because of the eggs being relatively larger targets. They can be hit by sperms fairly well. See, the spermatozoa will be released into the external environment, and we have already studied about the detailed structure of spermatozoa, which is having a head, neck, and a very flexible whip-like tail, with the help of which the spermatozoa can swim. So, a relatively large number of spermatozoa, in millions and millions, the spermatozoa will be produced and they will be released, and they will be swimming randomly in the water, trying to find out their ovum. And the moment they encounter the ovum, they fertilize the ovum, and the new generation will begin. The next type of fertilization is the internal fertilization. In many oviparous, which are egg-laying animals, viviparous, which are animals giving birth to the young ones directly, and oviviparous, which lay the egg but they don't, expel it as egg. They retain the egg in their uterus or some other structures which are meant for that inside the body of the female, and the egg will hatch inside the body of the female, and the miniatures of adults, the young one, will come out of the body. This kind of behavior is called ovoviviparity. In all these three forms of animals, the eggs are completely enclosed in an impermeable egg membrane, or they are retained within the maternal body throughout the development. In all such cases, the spermatozoa are delivered internally into the body of the female by some type of copulatory mechanism or by an intromittent organ of the male. See, if the fertilization is internal, then somehow the spermatozoa must be introduced into the reproductive tract of the female with the aid of some kind of organ which helps to transmit these spermatozoa from the male's body into the reproductive tract of the female. In such forms, the fertilization may occur in the lower part of the oviduct. For an example, in urodales, the salamanders are the examples, or in the upper portion of the oviduct, example, many birds and mammals, and or in the ovarian follicles as well as in the viviparous fishes such as Gambusia finis and many other eutherian mammals. So this is how the fertilization, the internal fertilization takes place. So let us assume this as a typical uterus, and in some animals the fertilization may occur in the lower part of the oviduct. Consider this as oviduct, okay, or I will draw a simple diagram of the oviduct. Let us say this is the ovary and the oviduct is this one, okay. And now this is the lower portion of the oviduct in many of the salamanders. This is the location where the fertilization takes place. In mammals and other animals, you can see the fertilization taking place in this region. And if you observe the human fertilization, it takes place in this region, and this part is called the ampulla of the fallopian tube, and this is the region where the fertilization takes place. So once we study about the types of fertilization, let us now look into the next topic, the approximation of the gametes. The fertilization, a successful fertilization for that matter, requires the sex cells are close enough in the proximity to bind to one another. Thus both sperm and egg must travel from their original deposition location to approximate at the fallopian tube. Say for an example, in human beings, the ovum will be produced in the ovary, and this is how the fallopian tube will be. Okay, the fimbriae will be there, they collect the fine. So this is a rough diagram of uterus, let us see. And the other horn will be on the other side, okay, like this. The fimbriae will be there, and then there will be ovary. So these ovaries are the location where the ovum synthesis will be there, the ovum will be produced, and the spermatozoa comes from here, right? The vagina will be open, so from where the spermatozoa will come and it will swim like this. From here they will swim and swim and swim and swim and reach the ampulla region. And the ovum will be somewhere here, and it will be caught by this fimbriae, and that will reach the location. Okay, this is the location where they meet, the ampulla region. So in order to achieve successful fertilization, both the gametes, right, the spermatozoa must move from here and the ovum must move from here. From their original place of production, they have to move towards a particular place where the fertilization can take place. So after the spermatozoon deposition, the prostaglandin present in the semen along with the oxytocin release during the coital reflex leads to the uterine contraction. The mixture of sperms with the secretion of uterine glands is passively aspirated. That means the spermatozoa, when it is being deposited here by the male through sexual intercourse, the prostaglandins, a kind of hormones, okay, which are present in the semen, along with the oxytocin, another hormone which is released during the coital reflex, that leads to the contraction of uterus. Now what happens when the uterus is contracted? The mixture of sperm, that means the sperm present in the semen, will be passively aspirated. That means that will be aspirated towards the uterine horns like this. It is worth remembering that the active motility of sperm does not help. Most sperm die within 24 hours of their release, as the spermatozoa gradually reduce in number by the barriers provided by abrupt constriction at the cervix and the uterine ostium. Of the hundreds of millions sperm emitted at a single ejaculation, only 1% enter the uterus. A trip from cervix to the oviduct takes from 30 minutes to 6 days. The oocyte travels via ciliary beads in rhythmical contractions with the musculature of uterine tubes. Finally, the sperm and ova reach the ampulla of fallopian tube. So merely the forward wriggling movement of the spermatozoa will not help the movement of spermatozoa from the vagina towards the ampulla. There will be other aids such as the rhythmical contractions of the musculature of the uterine tubes. It also helps to make the spermatozoa more reachable to the ampulla region as well. The fertilizing and anti-fertilizing reaction. The cause of agglutination of spermatozoa was studied by Lilly in 1999. While studying that, he observed the fertilizing and anti-fertilizing occur in the eggs and sperms respectively. They are directly involved in the reaction between the egg and spermatozoa. The main source of fertilizing is the egg itself and is located in the plasma membrane of the ovum. However, in the eggs of sea urchin and other echinoderms, it is produced by the layer of jelly surrounding the eggs and becomes accumulated in the external gelatinous coat. So this fertilizing and anti-fertilizing acts as a very important mechanism where a spermatozoa from a particular species can only fertilize the ovum of the same species. In the previous semester, while studying about the mechanism of reproductive isolation, we have studied in detail about the gametic isolation. The gametic isolation is established by this fertilizing and anti-fertilizing, which are present over the spermatozoa of the ovum and spermatozoa respectively. Now these two are just like lock and key, or just like two compatible blocks as shown in this cartoon. So these compatible blocks or lock and key will operate with high specificity so that the spermatozoa of a species is not able to fertilize the ovum of any other species. The fertilizing is a gel formed out of glycoprotein or mucopolysaccharide. Chemically, fertilizing is a glycoprotein or a mucopolysaccharide. As a protein, it contains a number of amino acids, and as a polysaccharide, it includes molecules of one or more monosaccharides. Monosaccharides such as glucose, fructose, fucose, or galactose are esterified by the sulfuric acid before it is coupled with the amino acids. So this is the chemistry of fertilizing. Okay, it is a glycoprotein or a mucopolysaccharide, which means a protein combined with a carbohydrate. Okay, so the protein unit of it will be having a lot of amino acids of different kinds, and different structures are obtained due to different arrangements of amino acids that you know the protein polymorphism can be possible by the differential arrangement of the amino acids. And then you know monosaccharides will be present along with that, and these monosaccharides are esterified by sulfuric acid. Both the amino acids and polysaccharides vary from one species to another species. That is why each species possesses a specific type of fertilizing. Molecules of fertilizing are quite large, and the molecular weight is around 3 lakh. And each molecule may have more than one active group, so that one fertilizing particle may become attached to 2 or more spermatozoa, thus binding them together. So you can see here, this is the fertilizing particle, and this is the spermatozoa coming. So each fertilizing particle have more than one active group or active site, so that to one fertilizing molecule many spermatozoa may be attached. Here in this case 4 spermatozoa may be attached. The surface layer of the spermatozoa, the plasma membrane of the sperm, contains another species specific acid protein called anti-fertilizing or anti-fertilizins. The anti-fertilizins can be extracted from the spermatozoa by heating, freezing and thawing or acidifying the water. The remarkable peculiarity of the fertilizins and anti-fertilizins is that they combine in a specific manner. That is, the egg fertilizing of a particular species reacts best with the sperm anti-fertilizing of the same species. No cross reaction occurs, no cross specifications occur during fertilization. And this is where the specificity of the fertilization is possible. Reactions between the species which are nearly related to each other are very much weaker. The reaction between the fertilizing and anti-fertilizing is very much similar to the reaction between the antigen and the antibody. In both these cases, a chemical lock is formed between two complementary substances. Thus, during capacitation, the details of this capacitation will be dealt in the 35th and 36th sessions of the same paper. Capacitation is nothing but the functional activation of the spermatozoa by the uterine environment. And during capacitation as well as the contact stage of the fertilization, when spermatozoa and eggs of the same species come in physical contact of each other, a chemical lock is established between anti-fertilizing molecules of the spermatozoa and fertilizing molecules of the unfertilized egg. And due to the fact many spermatozoa adhere to the surface of an unfertilized egg. It has been suggested that the main function of fertilizing anti-fertilizing is to thin out the number of spermatozoa around the egg, so that the chances of 2 or more spermatozoa fusing with the egg at the same time are diminished. So that's to avoid the polyspermia. This spermatozoa, anti-fertilizing and fertilizing will play a very important role. The next topic we are going to study in detail is the acrosome reaction and the penetration of the sperm. As mentioned earlier, except porifera and coelentrates, where the surface of the ripe egg is seldom naked, most animal eggs are enveloped by one or more egg membranes or gelatinous layers or follicular cells or both outside the plasma membrane. These layers constitute the barriers for penetration by spermatozoa and serve in preventing fertilization by more than one spermatozoa or by sperm of other species as well. So if you remember the structure of human ovum, you can see that at least 3 layers will be present outside the plasma membrane of the ovum. And sometimes it is also surrounded by the cells of granulosa cells. So this acts as a barrier for the entry of spermatozoa. If the spermatozoa has to enter into the ovum in order to fertilize the ovum, then somehow it has to penetrate through these layers. So how this penetration is achieved is nothing but the acrosome reaction and the penetration. When a spermatozoa is attached to the surface of the egg, you can see here it is now attached to the surface of the egg, it becomes motionless. So no propulsive movement will be there. Its penetration through the egg membranes and also through the plasma membrane of the egg is achieved by some physiochemical activity of the sperm acrosome. Certain enzymatic proteins called sperm lysins are produced presumably by sperm acrosome. The sperm lysins differ from one animal group to another animal group, further establishing the specificity. In some cases, the dissolution of the egg envelopes may be brought about by simpler means. Thus, it is believed that the jelly coat of echinoderm eggs may be dissolved as a result of acidification of seawater by carbon dioxide produced by the spermatozoa in the course of their respiration. In case of eggs with very thick and resistant envelopes, such as the egg envelopes of fishes and insects, the sperm cannot reach the egg at all points, but must penetrate through a special canal, the micropyle, left in the egg envelope of the chorion. See, if the wall of the ovum, so this is not the ovum of the egg, ovum of the insects or fishes, but anyhow, it will be like this itself, a plasma membrane, a nucleus, everything will be there with cortical granules. Now, if the surrounding layers of the egg is too hard to penetrate or too thick to penetrate, for an example, the eggs of fish and insects as well, there will be a aperture through which the spermatozoa can enter, and that aperture is called micropyle. So while studying about the eggs of insects, we have studied about this micropyle. It's an opening from where the spermatozoa can enter. That is the only place where the spermatozoa can enter into the ovum in fishes and insects. But in mammals, when the eggs are released from the ovary, they are commonly encased in a layer of follicular cells called corona radiata. See, this is the ovum now. Fine. Surrounding the ovum, you can see a single layer of cells, and this layer is called corona radiata, and it is composed of the follicular cells. These cells are held together by an adhesive cementing substance called hyaluronic acid. You can see here, this is the chemical structure of the hyaluronic acid. And this hyaluronic acid is a mucopolysaccharide. The corona radiata now acts as a barrier through which the spermatozoa must first penetrate to reach the plasma membrane of the egg. See, the sperm will come from these directions, any direction it may come, from here, anyway. But before they can enter into the plasma membrane of, this is the plasma membrane now, before it can reach here, it has to penetrate through this corona radiata layer, right? That is very important. Now this corona radiata is bound tightly by a substance called hyaluronic acid. Now if the spermatozoa has to enter, then it has to somehow dissolve this hyaluronic acid so that the cells which are attached adhere to one another may be released. Well, let us see how this is achieved. Now there will be an enzyme called hyaluronidase, and this is the enzyme which will be present in the granules of the acrosome, which will be released immediately after the attachment of spermatozoa to the ovum, and this reaction is called acrosome reaction. When this hyaluronidase enzyme is released, it has the ability to dissolve these bonds, peptide bonds that are, sorry, not the peptide bonds, the bonds in between the two monomeric units of the hyaluronic acid. So this bond will be now hydrolyzed and the molecule will be broken down like this. You can see here it is bound by a bond here, and while hyaluronidase enzyme acts here, it hydrolyzes here, it breaks the bond here, releasing two different monomeric units. For this purpose, the sperm's acrosome produce an enzyme hyaluronidase which serves to dissolve the adhesive and disperse the cells of the corona radiata. So when hyaluronic acid is broken down, the cells which are tightly bound will be now just removed. The breaking of the membranous barrier is not only mediated by lytic agents provided by the acrosome of the spermatozoa, but also acrosome itself undergoes morphological changes and forms acrosomal filament which help the sperm penetration into the egg interior. You can see here not only the acrosome is bursted out, releasing the enzymes, it also produces some processes called acrosomal processes. Detailed illustrations you can see in the next slide, I hope. Yes, you can see here these are called acrosomal processes, okay, long structures. And here also you can see a long lengthy structure is produced which is called acrosome process. So not only the dissolution of the substances in between the corona radiata cells, but also the production of acrosomal process is also the important process involved in fertilization. Next, the acrosome will burst. The apex of the acrosome bursts so that the membrane of the sperm and acrosome open apically and consequently expose the interior of the acrosomal vesicle to the outside. The two membranes join around the margin of the opening. So here you can see here this plasma membrane of the spermatozoa and the ovum will now merge with one another, join there, and then they release the lytic enzymes. The acrosomal granule is released and comes in contact with the egg envelope. It contains lytic enzymes which make passage through the egg envelopes. Therefore, shortly after its release, the acrosomal granule disintegrates and disappears. Now, shallow depression of the acrosomal membrane close to the nucleus now deepens and soon lengthens into a long slender acrosomal tube. As you can see here in this photograph, it's a long slender acrosomal tube is formed. Fine. Once the enzyme is released, the membrane will be dissolved. Now acrosomal process is formed. Fine. And it lengthens. Now the tubule becomes twice as long as the sperm nucleus. So the size of this tubule will be almost double the length of the nucleus of the spermatozoa. Now rest of the acrosomal membrane everts and is added to the acrosomal tubule at the base. This is simply an unfolding of the already continuous membrane. Now at the end, the acrosomal tubule gradually enters through the passage of the egg envelope like this. Okay, gradually it passes through here, and which has been made previously by acrosomal lytic enzymes. So this will be, this passage will be done by the enzymes that are released before by the acrosome. So through this they simply move. You can see here this is the acrosomal process, and the plasma membrane of the head of the spermatozoa now trying to move inside, and ultimately touches and fuses with the plasma membrane. Okay, so when it reaches here, it fuses with the plasma membrane of the ovum here. Now this is the plasma membrane of the ovum. Ultimately now it is fusing itself with the plasma membrane of the egg. Next, the sperm contents will be passed in. The fertilization cone protrudes from the egg and engulfs the acrosome tubule. The nucleus of the spermatozoon is drawn out towards the fertilization cone. The acrosomal tubule dissolves. The elongated nucleus along with the middle piece of the spermatozoon is engulfed into the egg cytoplasm. In other animals, similar types of events have been observed during the penetration of sperm into the egg envelope. Before penetrating the egg contents, and before activating the egg for further developmental events, the spermatozoa of many animals themselves get activated, like in the spermatozoa of the sacroplasm. This is the different types of fertilization and the behavior of the spermatozoa, and we are not going to focus much on this part because this is the comparative account of the sperm entry. But please remember here, the fertilization cone is produced by the ovum. A cone shaped structure will be produced by the ovum, and it engulfs the acrosomal tubule. Engulfing means it takes up the acrosomal tubule, and the nucleus of the spermatozoa is now taken inside along with the middle piece. Sometimes the entire spermatozoa may also enter. In some animals, the sperm activation includes rupturing of acrosome and formation of acrosomal filament or acrosomal tubules. However, the number and size of acrosomal tubule may vary from species to species. As in annelids, for an example, they have several acrosomal tubules. The mammalian spermatozoa, though possess acrosome, do not develop acrosome filaments. The spermatozoa appears to contact the surface of the egg by its lateral aspect. Following this action, the plasma membrane of the egg and the spermatozoa dissolve at the point of contact, and the spermatozoa is drawn into the interior of the egg. So this is the difference between the formation of acrosomal process in invertebrates and vertebrates, in particular the mammals. The acrosomal processes do not develop. The next part is the cortical reaction and the formation of fertilization membrane. Even before the fertilization cone is formed and the spermatozoa penetrates into the interior of the egg, a chain of physiochemical reactions is set in the egg cortex, that means inside the ovum. All these reactions are collectively called as cortical reactions. The reactions, the physiochemical reactions that are taking place in the cortex of the ovum is called cortical reactions. These reactions may differ from one group of animal to another, but in most groups, the cortical reaction lead to the formation of fertilization membrane outside the egg plasma membrane. This membrane blocks the entry of the late arriving spermatozoa in the egg interior and thus avoids polyspermy. The process of cortical reaction and fertilization membrane formation in different group of animal may be different, but anyhow they do offer protection against the polyspermia. Say in sea urchin for an example, as soon as the apical end of the acrosomal tubule touches the surface of the egg from the outside of the contact, a wave-like color change from yellow to white under dark field microscopy, if you observe it, travels rapidly around the egg cortex and is shortly followed by the elevation of fertilization cone from the egg surface and the formation of fertilization membrane around the egg plasma membrane. Electron micrographs of the sea urchins unfertilized eggs show that egg cortex is bound by two membranes, an outer 30 angstrom thick vitelline membrane and an inner 60 angstrom thick plasma membrane. Beneath the plasma membrane occurs a layer of cortical granules. The detailed structure of these cortical granules is already studied in the previous sessions while studying about the detailed structure of ovum of different animals. Now these cortical granules are present just below the plasma membranes, ready to burst out so that fertilization membrane is formed outside the plasma membrane in order to avoid polyspermia. The outer vitelline membrane separated from the plasma membrane undergoes expansion and becomes the outer layer of the fertilization membrane. The cortical granules explode and release the following three components: a darker, denser, lamellar and folded parts of the granules. These lamellar bodies unfold and fuse with the inner side of the already elevating membrane, the vitelline membrane. Globules which fuse together and build up a new surface of the viscous hyaline layer just at the outer side of the egg plasma membrane. The hyaline layer adheres closely to the surface of the egg, and during cleavage it helps to keep the blastomeres together. The liquid component of the cortical granules fills the pre-vitelline space between the new egg surface and now the completed and elevated fertilization membrane. It contains mucopolysaccharides and abundant water. All these structures, namely the vitelline membrane and the contents of cortical granules, thus form a fertilization membrane, which is much thicker, about 900 angstrom, and are very strong. Among other invertebrates, fertilization membrane formation has been observed only in certain annelids, such as in nereid. In others, no structural but metabolic changes have been observed in the egg cortex. In vertebrates, the changes which occur in the cortex are similar to the sea urchins. In bony fishes and frog, the cortical granules are broken down immediately after sperm's penetration into the egg cytoplasm. Their contents become liquefied and extruded on the surface of the plasma membrane of the egg and fill the pre-vitelline space occurring in between the chorion and plasma membrane and in between the vitelline membrane and egg plasma membrane in the frog. In both cases, the vitelline membrane or chorion itself does not transform into the fertilization membrane as in sea urchins. The chorion in fishes becomes hardened or tanned after fertilization, and no new membrane is formed in either of the two animals. In some mammals, such as man and rabbit, the cortical granules burst and release their content into the pre-vitelline space created between the egg plasma, lemma, and zona pellucida. Okay, if you can see here, this is the plasma membrane of the ovum, and this is the zona pellucida. Now, in between these two, you can see a space which is called pre-vitelline space. And now the cortical granules are released into this pre-vitelline space. In neuroidal amphibians and some mammals which lack cortical granules, neither any cortical reaction nor fertilization membrane formation occurs. So this is the detailed discussion on cortical reaction, approximation of gametes, and reaction between fertilizing and anti-fertilizings. For any further information about these topics, I would recommend you to refer these textbooks. Thank you.